Our final presentation is on topics in cultural evolution and their relation to the emergence of neurodegenerative diseases in humans.
In one of the books we read called Genes, Mind, and Culture, the authors outlined 3 different approaches to studying the transmission of brain characteristics. One of them is purely genetic. In this kind of transmission, changes in mind and brain are uncorrelated with changes in the social and cultural environment. On the other hand, there's pure cultural transmission, where differences among individual minds are fully correlated with matching differences in social and cultural upbringing. In the third one, which is a combination of the previous two, called gene cultural transmission, some inputs have a greater likelihood to have an effect on the mind and brain development, and thus receive more attention from the developing mind. Transmission is generally informed by factors such as perception, culture, language, reproduction, and others.
To get a historical understanding of the influence of culture in the evolution of the brain, we looked at a book by S. Milton called The Prehistory of the Mind, in which he argues that culture fundamentally changed the architecture of the brain by forcing humans to make associations between different types of intelligence that were previously compartmentalized. This change is mainly reflected in stronger functional connectivity between the prefrontal cortex and the rest of the brain. In turn, reconfiguration of neural networks allowed for the development of language and other cultural symbols such as art. One researcher notes that the neuropsychological functions that create the capacity for culture are very much akin to those capacities attributed to executive function, such as inhibition, self-awareness, self-regulation, imitation, and vicarious learning. With the advent of epigenetics, many theories supporting cultural transmission started to gain notice. Here we talk about 3 of them: the meme theory, reification rules, and Darwinian algorithms. All of them essentially cover what are known as cognitive programs that serve to support adaptive functions such as face recognition or more efficient decision making. We also looked at some of the ways that culture influences synaptic development across the lifespan. For example, human infants have a dominant right hemisphere, but research has shown that adults tend to have a dominant left hemisphere. There's also a shift from fluid to crystallized intelligence, and there is a type of neuronal Darwinism in which infants have an overgrowth of neurons and synapses, but across the lifespan there's pruning according to experience and assigning values to rewards. Finally, there's neuroepigenetics and plasticity, which we will talk about more in the rest of this presentation.
The next section is devoted to explaining neurodegeneration and neurodegenerative diseases as caused by a recent period in human evolution. The format of the remainder of this presentation is that of an annotated bibliography. Each page of our slideshow is devoted to a different article, either detailing theories of neurodegeneration, evolutionary theories of the emergence of neurodegeneration in humans, or explaining clinical aspects of specific neurodegenerative diseases in light of these theories. Instead of going into detail on each one, we will be glossing over the main points, extracting the essence with special attention to topics already covered in class, namely cultural selection, gene-culture coevolution, and brain plasticity. Neurodegenerative diseases are characterized by the atrophy, loss of function, or cell death of neurons in specific regions of the brain. Our first article expands upon this definition and puts it in an evolutionary light. The authors look at a recent period in human evolution characterized by numerous structural developments in the prefrontal lobe of the human brain that led to what we now broadly refer to as intelligence. They argue that neurodegenerative disease is the opposite of the development in those precise and extremely plastic areas of the brain. So those suffering from neurodegenerative diseases retrograde in evolution, opposite to what we observe in fetuses who go through the stages of Darwinian evolution.
Our second article qualifies neurodegeneration as an evolutionary genetic trade-off with cancer. The article analyzes cancer and neurodegeneration as diametrically opposed human disease types where neurodegeneration constitutes increased cell death, while carcinogenesis is defined by uncontrolled cell proliferation. Evidence for their theory is found in specific genes and molecular pathways, as well as opposite gene expression, and also diametrically opposed epidemiological links between cancer and neurodegeneration. We're now going to focus on different evolutionary theories of neurodegeneration.
The first article of this section, called Evolution of Neurodegeneration, asks why pathogenic alleles persist in the population despite appearing to be an evolutionary disadvantage. The disadvantage being that those versions of genes either code for certain proteins that accumulate and are insoluble in neurons that survive cell death, or rather, when expressed, directly lead to cell death, and are a cause of neurodegenerative disease in the human brain. The argument made by the authors is that genes associated with neurodegeneration have been selected for because of beneficial properties. Therefore, neurodegeneration appears to primarily be a byproduct, one initiated in the brains of aged populations where chronic stress and other stochastic processes can influence their behavior. This article considers the diathesis stress model for the genetics of psychopathology, which claims that individuals with certain genes may possess greater vulnerability to dysfunction when exposed to adverse environmental conditions than individuals lacking those same genes. But this goes back to the question that the previous article was asking: why do genes that code for a vulnerability or a disadvantage persist in the population? The article reconceptualizes this theory, positing that rather than vulnerability, the genes confer a greater susceptibility to plasticity. So the genes can present a benefit in positive environmental conditions and a detriment in negative environmental conditions. The final two studies of this section put forth a theory for the persistence of our susceptibility to neurodegeneration that is grounded in the idea of cultural selection. The authors observe that in Darwinian evolution, detrimental factors such as vulnerability to neurodegenerative disease should be eliminated from a species' genome over time, but this is not the case with neurodegeneration. They argue instead that the normal evolutionary process, natural selection, was supplanted by cultural selection in the form of self-domestication. The changes that characterized recent hominins were introduced through the rise of culture, and therefore the brain developments that have been observed over the past 40,000 years and the intelligence that they entailed were selected with susceptibility to neurodegenerative disease as a price to pay for these new valued cognitive functions. Finally, we're going to see how these different theories manifest themselves in the evaluation of specific neurodegenerative diseases, specifically Parkinson's disease, ALS, and schizophrenia. Our first close look at a neurodegenerative disease will be Parkinson's disease, specifically how the evolutionary process may explain in part our vulnerability to it. It's first important for us to note that Parkinson's involves the dopaminergic neurons that are required to regulate complex motor behaviors, and that these neurons are particularly prone to oxidative stress and, as a result, neurodegeneration. The article argues that the increasing prevalence of Parkinson's disease may result from a reduction in exercise at the societal level. Our genome remains adapted for the physical way of life lived by our hunter-gatherer ancestors, which was characterized by a high daily energy expenditure. Meanwhile, technological advances such as self-domestication and the digital revolution more recently has reduced physical exercise in the average way of life. The brain release of neurotropic factors, however, which support the growth and survival of neurons, is somewhat exercise-related. Thus, as exercise diminishes at the societal level, so does the release of neurotropic factors, and hence the growth and survival of neurons, and we end up with an increased rate of neurodegenerative disorders on the societal level. Our second example of a neurodegenerative disease is amyotrophic lateral sclerosis, more commonly known as ALS. The
The thesis of this article is that the clinical features of ALS could be largely explained by a failure of neocortical circuitry. This is because over the 5 to 6 million years of hominid evolution, the acquisition of enormously big brains with advanced cognitive ability, complex vocal organs, bipedalism, and opposable thumbs induced major neocortical changes with the new cortical areas and increased interconnections. The associated increase in complex connectivity and its increased spatial territory adds considerably to the metabolic demand of all the structures involved. As a result, they're more prone to internal and external toxic events, and mutations in neurodegeneration could be considered an unfortunate byproduct of these developments. Those neocortical structures selectively degenerate in aged humans and underlie ALS. The final article in this bibliography is an experimental study of adaptive evolution of genes underlying schizophrenia. The authors start from the hypothesis that schizotypal cognition is associated with creativity and divergent thinking, and that, as a result, schizophrenia may have evolved and been maintained as a maladaptive byproduct of recent positive selection and adaptive evolution in humans. Their approach was to do selective sweeps in human populations and maximum likelihood tests for selection during primate evolution over 76 genes. 28 of those were demonstrated to mediate liability to schizophrenia in either one or both experimental approaches. In the end, they concluded that, consistent with the hypothesis, schizophrenia represents in part a maladaptive byproduct of adaptive changes during human evolution. We can see how this analysis bodes well when compared to the example of ALS, which was deemed to be essentially a maladaptive byproduct of neocortical evolution. It also corroborates the third explored evolutionary theory of neurodegeneration put forth in this bibliography, where neurodegeneration is regarded as merely a price to pay for the increased intelligence that results from new structures that appeared in the brain as a result of hominization.